Assalamualaikum warahmatullahi wabarakatuh. My name is Ami Ashraf. Today I want to explain the experiment of bacterial identification. So there are 4 basic steps, which is prepare a sample from a patient and isolate whole bacterial DNA, make many copies of the desired piece of DNA, sequence the DNA, and also analyze the sequence and identify the bacteria. Okay, for the part 1 is sample preparation. So first, we should put on a pair of rubber gloves and we can start the experiment. So first, click the wire loop to continue, and then click one of the bacteria colonies in the dish. Next, click the microcentrifuge tube to transfer the bacteria colony from the wire loop into the microcentrifuge tube. Now, we will add digestive enzymes to our sample. Click on the yellow micropipette to continue. The microcentrifuge tube must sit for several hours. Next, click the microcentrifuge tube to continue. After several hours of enzymatic digestion, your sample is ready. Click the microcentrifuge tube to hit inactive the digestive enzymes. The digestive enzymes are now denatured and inactivated. Next, click the microcentrifuge tube to continue. Now, we will spin down cellular debris for removal from our sample. Click the centrifuge lid to continue. Centrifugation is complete. Unwanted debris have now been spun down. DNA we want is in the supernatant. Click the microcentrifuge tube to continue. The sample preparation is complete. Click the orange tray to continue to part 2, PCR amplification. The next part is PCR amplification. We will now add the PCR master mix solution to all tubes. Click the red cap master mix bottle to continue. Now, we need to set up the positive control reaction. Click the green cap positive control DNA bottle to continue. For the negative control reaction, we will add water. Click the blue cap deionized water bottle to continue. Next, click the PCR machine to open the loading compartment and load the PCR tubes. The PCR tubes have successfully been loaded into the PCR machine. Click the PCR machine to continue. The PCR amplification is complete. Click the orange tree to continue the PCR product purification process. The next part is purify PCR product. First, we will set up the microconcentrator column. Click the yellow cap of a bottle to continue. Click the orange tree to continue. The tubes containing the PCR sample, positive control and negative control, will be put on ice. Click the orange tray to continue. Click the orange tray to load the column into the centrifuge. Click the centrifuge to continue. DNA is now trapped in the column. Now, we need to loosen it into a new tube. Click the orange tray to continue. Click the inverted column to continue. The first collection tube is no longer needed. Click the orange tray to discard it. Now, we will centrifuge the assembly to collect the DNA into the collection tube. Click the orange tray to continue. Click the centrifuge to continue. DNA has now been collected in the collection tube. The column is no longer needed and will be discarded. Click the orange tree. The PCR product has been purified. Click the orange tray to continue to part 4 DNA sequencing. The next part is prepare for sequencing. First, we will dilute the PCR product to increase the volume. Click the blue cap distilled water bottle to continue. The green and blue strip tubes already contain PCR reaction mixtures in them. Click the strip tubes to continue. The strip tubes are ready for PCR amplification. Click the PCR machine to continue. The strip tubes have been loaded. Click the PCR machine to continue. The sequencing preparation is complete. Click the orange tray to continue to part 5 DNA sequencing. The next part is DNA sequencing. We will now load the automatic sequencer. Click the orange tray to continue. Click the DNA sequencing machine to open the doors and load the tray. Click the orange tray to load it into the DNA sequencing machine. Click the inner gray door above the orange tray to view the DNA sequencing animation. You have completed the DNA sequencing section. Click the orange tray to continue to part 6 sequencing analysis. The last part is part 6 DNA sequence analysis. So follow the instruction in the notebook window. Click the computer screen when you are ready to identify the bacteria. So we just click the computer screen and identify the bacteria. So the correct bacteria is C. Bartonella henselae. That's all for today. Thank you.